but it's actually not the first one that evolved. This alternative pathway and the lectin pathway evolved much earlier in our evolution and are therefore actually more classical pathways than the classical pathway, but whatever, however you want to remember these. So the classical pathway is a pathway where our complement system will bind to antibodies that have recognized antigen and are now accessible, and these are going to bind to IgGs and IgMs, not the other antibodies, and actually they bind at a much higher rate on IgMs versus IgGs, like 1,000-fold more. But so once they bind, then they will act on. So you have C1, and then again we have these MASPs that are these R2 and S2. We have two R's, so C1R. We have two of those. C1R, these are the blue guys, and C1S. We have two of those, and those are the ones that have the activity where they will now break down C4. So remember, our order was C1, then C4, even though they kind of got the C2 over the top. So then C4 is broken down into C4A and C4B. So C4B is the larger subunit. C4A goes and it now has inflammatory. It causes inflammation, influx of cells. So now half of the portion of that has that type of inflammatory activity, whereas the other portion comes and creates what's called the C3 convertase. So C3 convertase doesn't necessarily have C3 in it, but it means it's going to now convert C3 to its active state of C3A and C3B. And so C3, out of all of the complement, C3 is kind of the linchpin or the keystone to this cascade. Once you convert C3 into C3A and C3B, now you're going to get really the effects of the cascade are going to cause opsonization. So it's actually C3B is going to go and it's going to coat cells, or the C3A is going to really drive inflammation. And then C3B also will bind to this C4B2A complex and will change it into a C5 convertase. All that means, again, is that that's going to convert C5. So C3 is involved in the opsonization, so making the cell tasty. And then C5, and then it's also going to contribute, so you can see how C3 is actually in this cycle, this basically magnification cycle where C3b will then also bind to cell. So all of this is happening down to a cell, all of this amplification part. So you have this amplification loop where C3b is going to bind to a cell as well now. It's going to bind to this BB portion that is part of this alternative pathway. But this alternative pathway is kind of always occurring in small numbers. And if for some reason C3b is bound to a cell, it will then bind to small levels of activated B, lowercase b, that is in this system, and it will also form a C3 convertase. So there's more than one C3 convertase. Here we have two examples, the C3b and BB, big B, little b, and then we also have the C4b2A. And so you'll see that both the lectin pathway and classical pathway the end result is making this C3 convertase.
So what's the difference between C3b,
these guys?
C3b, yeah.
They have the same it's the same end result. They have different subunits, but the same enzymatic activity. So they're both C3 convertases. All you need to know is that all of these pathways lead to a C3 convertase. So if you know that classical pathway, a good question would be, what what's the common goal? What's the common end result of all three pathways? You would say, well, they're all going to form a C3 convertase. And now the pathway is the same from there on out. So these two pathways, the classical pathway and lectin pathway. So a lectin pathway is now where the actual, this mannose binding lectin. So mannose is a sugar that is on bacteria. It's common sugar on the surface of bacteria. We don't have mannose on our cells, so that's a good target. So here's another one of those innate targets on bacterial cells that we've adapted to have receptors that are specific for mannose. Just any mannose that sees this mannose binding lectin is going to bind to that carbohydrate on the cell surface of a bacteria or whatever, and some viruses have HIV, I think, has mannose. RSV has it. So there are different, lots of pathogens have these targets where these MBLs are going to bind to them. So as opposed to here, we have the C1 complex binding to antibodies. Here now, so this is another good question is, which of these pathways is involved in the adaptive immune response? That would be the classical pathway, as opposed to the lectin and alternative pathway are going to be involved in more of an innate, these are innate responders. So these are things that kind of developed first in our evolution. And so this lectin binder binds to these carbohydrates and now becomes active and will do the same exact thing. It's going to break C4 into a smaller, into the C4A and C4B. That's going to then bind very quickly to the cell right next to it. And if it doesn't do that, it becomes inactive. And so that is it. Oh, different order. I'll move the order.
Oh.
Go 2 or 3 slides in. One more. Here you go. I changed the order because I thought it was easier to talk about it with pictures before I started throwing lists in there. So you have your it binds carbohydrates. It does the same thing. Now, because it's bound, it's going to change conformational shape enough to become active and have these now are going to break down C4 and also C2, and now you're going to get the formation of this C3 convertase. So now this C3 convertase will then convert C3 into C3A, C3B. C3B will then bind to the cell surface and form this other C3 convertase, which is C3B, and if there's any level of BB around, that'll form that convertase. And then it'll also go and it'll bind to C4B2A and create this C4B2A3B, which is a C5 convertase. So C3B is involved in opsonization, whereas now if we convert C5 into C5B and C5A, again, this smaller subunit has inflammatory activity, but this larger subunit now goes to form that the complex that's going to punch holes in the cell. So now this whole cascade is just going to now, everything goes downhill from here for whatever cell happened to have that initial complement event. So either because antibodies bound to it or because the MBL bound to the cell, or in this case, even the alternative pathway, this is all happening within, because we have lots of C3 floating around in our serum and our blood, circulating through our entire system. Small amounts of C3 are always being converted to an active form, which is C3H2O, and that will bind to BB, and that will then convert. That itself has actual, as you can see here, C3 convertase activity. So even now, not bound to a cell, it'll quickly, any cell that's near it, it may bind to it, create that convertase, and start converting C3. Now, we have because this has a half-life, very quick half-life, it's not going to stay stable unless it binds to another molecule called properdin, and however you want to pronounce that, properdin. That will stabilize that. So if that properdin, and properdin, much like this MBL, we found it has specificity to pathogen cells as opposed to our cells. And so it has this preference to bind to pathogenic cells, and therefore properdin can be a target for, can bind to this C3H2O, and so then it can stabilize it. And
what's a C3H2O?
it's a form of C3 that naturally becomes active. So it's kind of, this is constant, C3H2O is constantly forming and disassociating, and so you have this constant formation. So it's kind of it's constantly looking for things that it can stick to, and if it doesn't stick to something, it just converts back to C3.
So what's this C3H2O? Is it the active form of the C3?
Yeah, so it's a chemical change, it's a reversible chemical change that happens to C3.
Even worse.
Yeah, and I don't expect anybody to give me the organic chemistry on that, or the pathway. You just need to know that it's C3H2O is constantly forming, and it's part of this alternative pathway. So this would be an important kind of thing to kind of memorize, is just this formation of C3H2O, which
What's
has
BB then?
BB is then another factor that B is broken down to BB, and that also happens at very low levels. Naturally, it's not induced per se by an enzymatic event per se, but it's going to be just naturally, this is a naturally occurring but very unstable product. And so it only becomes stable when it binds to a cell.
BB is different from C3H2O.
Yes. BB is this little,
So
this
BB
blue, this
is
darker blue.
originated from factor B.
Yes. Yep, and that's where knowing the nomenclature makes it a little easier to kind of understand where these are coming from. So this will only become stable if it binds to a cell. It has a preference for not only does it have a preference for pathogenic cells, but it will, in low levels, every once in a while bind to our cells, but our cells have ways to break that down further. So we have enzymes that are built into our cells that are going to break that down. So that's going to be part of that protective pathway. And we have lots of redundancy to that pathway because it's kind of important. We also have things that will block it at this level, things that will block it at this level as well. At the C5, so we will block the formation of this MAC complex, and we'll talk about that in a second. So what are some of the components of the complement system? Again, we have this IgMs and IgGs are kind of your preliminary binders, something that'll bind to a pathogen. You have your MBL, or your mannose binding lectins or phycolins. There's actually a lot of different lectins and phycolins. So we mentioned mannose binding lectin because it's kind of an ideal one. It's kind of, we're going to use that as our example, but it binds to carbohydrates on the surface. And then these guys will then bind to C1, like for example, and C1, when we say C1, so you're going to see sometimes C1Q, and then C1R, C1S, there's two of these, two of each of these that bind to the C1Q. So that's, when we're talking about that, we're talking about C1. So this C1 complex is made up of these subunits. And they initiate the classical pathway to binding by binding IG. They bind to apoptotic, so they're also involved in binding to apoptotic blebs, so cells that are in the process of dying or being apoptotic, they'll bind to them and help to target them for phagocytosis. And these subunits are the ones that have this serine protease activity that's going to cleave C4 and C2. So C4 and C2 are the next guys down. So C4 first and then C2. And then you have these MASPs or MBL-associated serine proteases that are going to also associate with like the lectins. They're going to be like, they're going to have their serine protease activity, and they're going to cleave the C4 and C2. So on the for the classical pathway, you have this with serine, the C1R and C1S have serine protease cleaving activity, but on the lectin, so you're going to have the MASP1, the MASP2, which has the serine protease for the same targets, C2 and C4. So C2 and C2 now is going to be broken down, cleaved by these guys now, we're going down, and into A and B. So A is going to have serine protease with C1 and C4, and is a C3 convertase to form that C3 convertase, and then this is going to be inactive in the complement pathway. C4 now, it's the B subunit that's going to bind on the cell membrane with C1 and C2A and to form that C3 convertase. So these are the guys that are going to make up that C3 convertase. Again, that C3 convertase is the end result of all of these pathways. And it's going to be made up of these subunits, or this subunit. And then you get your sort of C3 again. And when I say this is kind of the key pin, because actually, in a lot of what we know about these systems and why they're important are when they are defective in people. And so people who have any defect, especially in this portion of the pathway, have real problems. They have a lot of immune problems where they're clearing cells they don't want to. You have like IPP, you have clearing of platelets, you have clearing of red blood cells, anemia, so it's really, really dangerous to not, you have increased risk of infection, stuff like that. So this pathway can, you can have problems, a lot of problems, depending on if you have mutations in this part of the pathway. So C3 is then broken down into first, initially C3A and C3B, but it can also form, like I mentioned, C3H2O is there's this internal thioester bond that undergoes hydrolysis, and that's what forms that C3H2O. And that happens naturally, and it's a very unstable. But it can be broken down even further. So this is, and then we have our factor B. So that, again, you also saw in that alternative pathway, factor B binds to C3H2O and then is cleaved by factor D into two fragments, BA and BD, and then that smaller fragment, it inhibits proliferation of activated. So they all have kind of this, nothing's wasted. BB, that larger fragment, forms basically that C3 convertase. So now the C3 convertase from the alternative pathway is formed by this C3 B and BB. So let's see, what else do we have? Then we have C5. Now, so C5 now, we have a C5 convertase that is formed by another C3 subunit binding to that C3 convertase, and now the C5 convertase converts C5 to C5A and B, and these will form this MAC, so membrane attack complex. So that's that complex that punches holes into cells, and it's going to be made up of C5A or C5B, which is going to bind to C6. C7, C8, C9, and then the rest of C9 is going to make up that large, basically the hole that's going to form in the membrane of whatever cell it's attacking. let's cover the classical pathway. We're kind of going backward a little bit, but again, the classical pathway is initiated by an antibody binding, so IgM or IgG binds, and they to an antigen, and then this allows for the binding of C1q. So the C1q is going to bind, and here it's shown bound to two antibodies, which is really important because it will not become active unless it's binding to two or more antibodies. And so binding to one antibody doesn't work, binding to two works. And this is where actually why IgM has a higher specificity, because IgM has many more of the binding portion for this. And when IgM forms these rings, and it looks kind of like a flower or a star when it forms, and so the portion, these tails are sticking out. And if you get C1q bound to one of these tails, if it's inactive, if it's not bound to antigen, they won't ever touch two of them because each one is sticking out and they can't reach two of them. But when that antibody IgM binds to its antigens, cognate antigen, it actually turns into what's called the staple form. All the tails kind of point downward like feet. Like instead of being like this, they point down like this, and now they are accessible C1q because they're closer together, and now C1q can bind to two of them, become activated, and start that cascade. And so, IgM has a lower affinity, let's say, to binding to two, because you have to have a lot more IgMs bound in closer proximity to be able to get activated complement. And so we have this portion of complement. It's like this collagen-like triple helix, binds to the CH2 domain of an antibody. And here's a picture of that. So C1q binds to only antigen-bound antibodies, because we don't want it becoming activated just on antibodies floating around, because we have plenty of them. It induces conformational change in the C1R molecule, activating it, then the C1R, then activates the second C1R, and the two, and then the two. So it's all this cascade again. Once the conformational change of C1q happens, it activates C1R, C1R activates the other one, then it activates C1s, and then C1s is the molecule that has that activity that is going to cleave now. So C1s cleaves C4 and C2. C4 is cleaved first. So remember, you're going from C1, C4, C2, and the C4b binds to the membrane close to C1. So again, if it doesn't, it's got a very short half-life, so it's only going to be able to bind to the membrane closest to wherever that complement is bound. It's not going to float off and bind to your own cell or a non-infected cell. So it's very short-lived and it will bind. And most of them don't, but some of them are going to bind to that membrane close to the C1. C4 binds C2, and then exposes it to the action of C1s. So that's why C4 comes first, because you need the C4b to bind, and then it will bind to C2 and expose it to the enzymatic activity of C1s, and then C1s cleaves the C2, creating the C3. And now you've got your C3 convertase, so, which is this C4b2a. Okay, so that's why the 4 comes first, because the 4 has to bind to the cell membrane, and then it binds to C2, kind of turning, let's just say, turning him over on his belly, allowing the protease to now cleave him. So the classical pathway is initiated, so that's what I just said. So how does that binding happen? It's a thioester bond, going all the way back to your organic chemistry roots, is exposed when C4 is cleaved. This bond is reactive binding to amino groups, so what our cell membrane is made up of lipids and lots of proteins, so the amino groups on the surface are targets. If no target is present, the bond is quickly hydrolyzed and rendered inert. So that's how we keep these guys from just binding to anybody, and so you get this thioester bond that binds to very quickly, and if it doesn't happen, they become hydrolyzed and now they can't bind. And so that's that. So it's a strong covalent bond. So it's gonna when it binds, it's gonna be strong, and then it's only gonna be broken down by an enzyme. And that's where our cells have to have enzymes. Then, if we accidentally form this complex, on our own cells, we need some enzyme to break that and get rid of that guy.
Wait, so a cell's going to produce an enzyme that's going to hydrolyze the C4B off of the membrane?
Our own cells, yes.
Okay.
So most of our own cells have those. So that's just one of our, they're all going to have that, and that's just going to kind of protect us from our cells. It's a lot of work, yeah. It's a lot of work being us, being mammals. We're constantly having to fight ourselves off. So the classical pathway is initiated by this antibody binding, and now the end result is this C3 convertase. Now C3, it cleaves many C3 proteins, and this is where this amplification happens. C3 gets cleaved into C3b and C3a. C3b will also bind and become a convertase itself. It will also bind to the initial C3 convertase and create a C5 convertase. So we're going to start out with the C3 convertase. And then C3 itself, C3b itself, is going to contribute to more and make more and more and more and cleave more C3, and basically it's this cascade event. And so once you get C5 convertase, then you're now going to, that C5 is going to get cleaved into C5b and C5a, and C5a will go off. And C5b will now be involved in producing that MAC.
Excuse me, can I ask you a question?
Yes.
What's the C4B2A?
So C4b2a is, so
That's the C3 convertase.
so remember C4, so when this C1 binds to an antibody, it's going to have enzymatic activity that breaks down C4 into C4a and C4b, and C4b is going to stick to C2, and it's going to change C2, it's going to allow C2 to get cleaved, and you're going to end up with C4b, this light blue,
Oh.
and C2A is the dark blue. So C2A, and this is now that C3 convertase. So these two subunits, 4 and 2, or 4B and 2A, are going to form that. So this guy is that C3 convertase. C3 convertase is C4B, and 2A is now it's now an enzyme that's going to break down C3 into C3b and C3a. So it's going to cleave that, and then that is also going to go and bind to that. Now you're gonna have this now C5 convertase, and now this guy right here is going to bind to C5, chew them up into C5a and C5b. C5b sticks the same way to the cell surface, and then it will then initiate and bind to C6, C7, C8. So it's really easy now that C9 and form the MAC, which is going to chew up the cells. Right, so that's a classical pathway. It's going to punch holes in it. It's a hole punch. So then we've got our lectin pathway. In this, there are many lectins. The one we're using as an example is going to be this mannose binding lectin. So in this lectin pathway, mannose binding lectins float around, it says, wow, here's mannose. That shouldn't be here. I think I'll stick around, hang on to this mannose. Same thing that happened, remember, with the C1. C1 binds to the antibody and changes its conformation so now that it has enzymatic activity. Well, same thing with the MBL. MBL binds, instead of the C1 R and S, it has these MASP1 and MASP2. MASP2 is the one with the enzymatic activity, just like the C1S. Is going to now do and the MASPs cleave C4. C4 binds to the C4B binds to the cell. It sticks to C2. It converts C2 to C2A, and now you've got your C3 converter. So there, you just learned another pathway, because the only difference is MBL is going to directly bind to a cell, to sugars on the cell. And so this guy is adaptive. So complement's involved in the adaptive immune response and in the innate immune response.
Oops.
Yes.
In the picture you showed us with the complement 1 binding around complement 1Q, is that the natural form, or is complement 1R just wrapped around it after it's met with the antigen bound?
Does it bind? Does it only bind? So you're asking if C1R only binds after it's bound, or is it always floating around together?
Yes.
Is that what you're asking? Yes. I don't know.
I guess
I'm not sure about that one. Look it up and let us know next week.
Extra credit for the test.
Yes.
For your efforts.
If you can teach me something, then you get extra credit. So now we have the alternative pathway. This one is happening not on a cell surface initially, even though here we're seeing the cell surface. So it has to, in order for it to become active and stay active, it does have to bind to a cell surface. But it starts all in the soluble form, in the serum. You get C3, which is going to naturally, so we have C3 floating around, lots of it, and you're going to get a natural form of C3 forms that is C3H2O, which is going to bind to factor B. Factor B is then going to have activity on factor D, and then that factor D is going to actually, well, okay, so factor D is going to bind to factor B and actually cleave B into BA and BB. So now you have C3H2O and BB. Now this naturally has, this is your C3 convertase here.
Which one is the BB?
BB?
Yeah.
This blue guy, dark blue. Teal is the C3H2O. I guess that's teal. I don't know what you call that, light blue. And then the darker blue or navy. I don't know. Actually, I don't know my colors. That's not my. Okay. But this guy's the BB. And so this is now also a C3 convertase, part of the alternative pathway. And then this C3 convertase is very unstable. So it actually, it can start to convert C3 into fluid phase, and then that will bind to but it happens very rapidly. And if there isn't a cell for this to stick to, then it's not gonna continue, because as you remember, C3b has to quickly bind to something, and then you get that will form, and then it will form more, and if it binds to the antibody, and this actually has to be stabilized. This complex has to be stabilized by another molecule to stay stable and continue on. And that other and so this is how this other molecule is where we're kind of the checks and balance to make sure that we're only binding to non-self cells, because this other molecule called piperidin, or factor P, let's call it. Okay, thank you. Now we got an easier pronunciation. Factor P will come and then stabilize that convertase. Now, it can either, there's two different ways this can happen. Piperidin can come in and bind to it, the complex, and stabilize it. Or piperidin can actually be bound to a cell already and that as that C3 convertase naturally forms, it will bind to preparedin that is already on the cell. And that's where a little bit of the specificity comes in that factor P preferentially binds to non-self cells, yes.
What is the end result for the alternative pathway of the lectin and
It's also the C3 convertase, and which drives C3, C5. So again, once we get to this C3 convertase, it's the same thing.
and the initiator for the alternative pathway, we did the lectin, which is the
So the initiator itself is actually, I guess you could kind of call preparedin an initiator. It stabilizes. So here you don't have like C1 or MBL. You don't have something that's going and targeting. You have this that's happening constantly at low levels, and kind of, I think it probably evolved because maybe, hey, I guess there's some pathogens out there that don't have these sugars that we recognize, or downregulated sugars. Maybe they haven't been made, and this evolved before antibodies were around, so let's not even think about that. So let's think about sugars and other targets. There were going to be pathogens that didn't have any of those targets.
The only reason why I asked is
Yeah.
I remember reading in the book that the only reason why this is called the alternative pathway or the classical pathway is the classical pathway was discovered first.
Yes.
So
Yeah. So alternative, yeah, probably not the best name for us to remember. This is the original pathway. This is I think the lectin may have evolved first, and then this one, as this one evolved, it was able to clear out, or maybe even this one evolved first. I don't know. I don't think they know actually. So you had a hand up? Somebody? I saw.
Yeah.
Yeah.
Different questions. So many.
Oh, yeah.
All right. So at what point? Are there more than one point where the C3 convertase and all preceding molecules are unstable? Or is this the part where it's unstable, just with the C3 convertase?
It's kind of unstable the whole time, but very low levels, it's forming complexes.
So it just keeps going, but unless it reaches the point where it reaches factor B,
Yes.
then it'll just reverse.
Yeah.
Okay.
So they call this the alternative, which is because this is part of the alternative pathway, tick over pathway, because it's, I guess, constantly happening and undoing and ticking over and over. I don't know who comes up with these names, but bless you. So this is another. So it can either bind and then preparedin comes and stabilizes it, or preparedin or factor P will bind to a cell. It can directly bind to a surface. This can then recruit C through B, which is going to naturally form in factor B. Again, these are naturally forming, not triggered by anything in particular. Factor B is recruited, cleaves factor B into BB, and you get now this C3 convertase from the alternative pathway. And then subsequent steps are identical. So again, if that becomes a stable complex here, then now that's going to just start the same pathway. Well, it's initiated in 3 different ways. Initiation, and then the third way is actually, so I talked about 2 ways. It could either bind to the cell and then preparedin comes and binds to it. Preparedin can bind to the cell, it binds to preparedin, or the alternative protease activated pathway is induced by thrombin. Okay, so this is now, we have a link between the complement system and our coagulation system. So let's say you have a wound and bacteria are coming in and you're starting to form, you have clotting factors that are coming in. Not only are the clotting factors going to stop you from bleeding out, but your clotting factors are actually supposed to stop the pathogens in their tracks, and they will now start to induce complement. So these guys are working all in conjunction, so you get clotting factors that are now slowing down pathogen influx, localizing neutralizing pathogen to a certain area and inducing complement within that area through the alternative pathway that's going to just start binding to every cell in the area. And we kind of just start throwing, it's like kind of shooting and just hoping that you're hitting people, but then hoping that the good guys have body armor and are able to not get killed. And our own cells are going to be fighting off that complement cascade as it's being formed on them. So, what are the results? So now we've talked about how can we induce C3 convertase. Okay, we're past that now. You got C3 convertase on the cell. It's there. What does it do? It contributes to C5 convertase. C5 convertase now. So C3 itself is going to be your opsin. It's going to make cells tasty. They're going to get phagocytosis, but it's also going to target cells for death. Probably why it evolved first too, because maybe we hadn't evolved phagocytic cells, so we needed a way to kill cells directly with this system. And so C5 then binds to C6, C6 binds to C7, C7 binds to C8, C9, and then C9 forms this nice big hole right in the middle of the membrane. Everything leaks out by myself. Yeah, by myself, because as you know, cells have membranes because for a reason. They're trying to keep certain concentrations of things in, certain out. It's that differential concentrations of cations, anions, and also contents within the cell. So once you lose that membrane integrity, the cell's going to die. And so that's the C5 creates this MAC or membrane attack complex. So C5 initiates the generation of this membrane attack complex. And that's this core structure disrupts osmotic integrity, resulting in something like that. So pretty simple, straightforward. So 3 main classes of complement activity. So here we've got, again, so lysis of bacterial cells in the cell membrane. We get opsonization, makes it tasty for phagocytes. And the byproducts of these, like 3A and all the little pieces coming off of these, are going to induce inflammation and chemotaxis and anaphylotoxins and bring things in. So this is all of what's happening. And it's these complements also this interface between innate and adaptive immunity, because it can augment antibody response, enhances immunological memory, it enhances antigen presentation as well. So these molecules also help with antigen presentation, and we'll talk about that more when we talk about antigen presentation. Potential effects on T cells. So these are the molecules. These also have effects on T cells. So this is evolved to really play a huge role in adaptive immunity, but also in the contraction phase of the immune response. They help clear, they can also be involved in clearing immune complexes from tissue that are, once they're done doing their job, clearing apoptotic cells. And they believe that these are actually some of the earlier functions of the complement system, and induction of regulatory T cells. So these molecules will actually help induce now a suppressive environment after they've gone in and done their job, or if they're mistakenly attacking our own cells, or if we want to get rid of cells we're done with, like red blood cells. So, I have not been looking at this. Okay. Honestly. Okay, so complement receptors, they connect complement tagged pathogens to effector cells. Receptors on the host cells allow for discrete and differentiated responses. So here are different receptors. So here's now the receptor end. And usually when we're talking about receptor, we have a little R in there somewhere, or a big R. So we have receptors for C1. So C1R will bind to C1. and these are on the surface of like macrophages, the eosinophils, these and lead to clearance of the immune complex. They enhance phagocytosis. So this is what the receptor. So if the receptor binds to these guys, these are what's going to happen. The receptor 2, C receptor 2, it's going to be its ligands. So this is what it's going to bind to. C3D and G. So these are further breakdown products of the C3B. Gets further broken down into D and G and C, and these enhance B cell activation. So further effects, receptor 3, okay, is going to be on B cells, and its ligands are going to be a few of these other, like factor H, we haven't really talked about, binding to adhesion molecules on leukocytes, facilitate all these other effects. So you can kind of go through and look at some of these. I'm trying to see if there are any that are really important. T cell activation, so these are the receptors that are going to do that. Granulation, down to C3A. So some of these are going to induce target cells are going to say, some of these are going to induce phagocytosis, whereas others are going to induce degranulation of the cell. For example, if this granulocyte sees its target of C3A, it's going to degranulate. So it's going to spit out its granules. They're going to be have all these lytic enzymes, and it's going to have all these peroxidase and peroxides, and it's just going to create that pussy, nasty environment that nobody likes. And so those are the different end results of binding to that complement. So it's good to know that not only can it lead to, so that's an important thing to know, not only does it lead to phagocytosis, but it can also lead to degranulation. So receptors, so these are important receptors, CR1 on leukocytes and erythrocytes. On red blood cells, they help to bring the immune complex to the liver for clearance. So the red blood cells, short-lived. We have to have a way of getting rid of them. Complement is a part of that. Complement will bind to those receptors and then lead those to the liver to be cleared out. On phagocytes, helps bind the complement-coated bacteria, enhance ingestion and destruction. Or on B cells, it helps to bind so when it binds to complement-coated antigens, it enhances ingestion for processing, not necessarily to just digest it, but to process it and present antigen now to the T cells. So each one of these cells, when it sees complement, it may do a different thing.
So this one is C1 receptor.
Yes, C1 receptor. These are the three functions depending on what cell it's on. Yeah.
I don't see it in my book written anywhere. Which complement were you talking about? Was it the CR1 that helps with degranulation of the
It was degranulation was C3 AR. So then also now we've talked about how it enhances because it binds and it can bind to an antibody. Antibody can then bind to an FC receptor. But here it also, so complement can actually, it can bind, so C3D, that's a further breakdown product, can actually bind to antigen. So if this is all happening, it'll bind in a certain area, you get C3D, further breakdown product of C3B, it will actually combine to antigen, and then that is a target for CD21, which will be on, so these, let's say this is your B cell, and so your B cell has your antibody here, your antigen, CD3 binds to that antigen, and then it will actually increase the is it affinity or avidity? Let's see if I can get it right. The avidity, see, the affinity is how strongly antigen binds. But now you're increasing the avidity, so you're making it even a stronger bond and getting even more activation of that B cell because complement is involved. So complement will actually increase the avidity of that event. Okay, so those byproducts, so we talked about what it does to the cell it binds to. And also you have C3A that floats off, C5A that floats off, and will bind to receptors, and then will induce an event. So these will induce inflammatory responses. They will bind to their receptor, which is starts this G protein cascade, which will lead to chemotaxis. So what does inflammation do? It brings cells to the area that's infected. It will bring in macrophages, so phagocytic cells, neutrophils, going to degranulate, basophils, degranulation. So these are phagocytic and degranulation, basophils degranulation, eosinophils degranulation, mast cells. So it's going to bring in all of these cells because it's going to basically start to secrete chemokines and basically cause this inflammatory or anaphylactic event, which is good in small doses, but then in larger doses can lead to anaphylaxis. Let's see, C3AR and C5AR on granulocytes are going to be stimulate release of more pro-inflammatory cytokine granules. So again, here's those degranulators that lead to degranulation. So let's see, complement enhances host defense. You've got these MAC-induced cell death, promotion of inflammation, promotion of opsonization. Here's just reviewing what we've been talking about. So coats cells, it leads to them being phagocytosed, or punch holes in cells, and lead to inflammation. It also clears cells, erythrocytes. It's going to MAC-induced cell death, so basically promotion of inflammation. Again, this is the same thing. Increases uptake of antigen by antigen-presenting cells, help provide secondary signals to B cells for activation, can stimulate T cells. So again, all the things kind of we read through when we were reading the specifics on a lot of those. And it clears, so again, it can clear cells that are done or dying. Lymphocytes are no longer required, so complement aids in the disposal of those apoptotic cells and bodies. So here's an image seen of the blood's coming off of that apoptotic cell, and C1's binding to the outer edge, and it aids in the removal and disposal of those cells. Now, how do we regulate? Regulation of complement. So again, that's great. We just don't want that to happen on our cells. So it's passively regulated by protein stability. So again, these are all short-lived. So we're regulating it by not having it float off and be systemic. And also cell surface composition. They're not meant to stick to our cells, even though they will. They just won't do it as well. So short half-life of C3 convertase unless stabilized by piperidine. Piperidine has a higher affinity for non-cell cells. Cell cells possess different carbohydrate structures that are more effectively bound by fluid-phase proteases. So we kind of will cover our cells in other proteins so that they aren't sticky for these guys. And these are more readily inactivated C3b through hydrolysis protecting cells. So that's another way. So here's some of the regulatory. So we have C1 inhibitors. So these are trying to keep things in check by just inhibiting it in the fluid phase, so it's not running around active. We have these DAFs, decay accelerating factors, and these are the pathways that they block. It accelerates dissociation of your C3 convertase. So C3 convertase is always being kind of broken down, and so we have DAF kind of associated with it's membrane bound. So our cells, almost all of our cells will express levels of DAF on the cell surface. So it's gonna, if that complex forms on our own cells, we're gonna get it off of there. C1R, receptors, so classical, alternative, it blocks these pathways. So you can go through and kind of see what are the different ways we block things. We block formation of or accelerate dissociation of C3 convertase. So a lot of these we have redundancy, so we're blocking that C3 convertase from forming. We're getting rid of it quickly. If it does form, further breaking down products that form it. Let's see, we bind it up and prevent insertion into the host membrane. We can also block formation of the C9. So hey, say we get that bound, we make some C5 convertase, we're starting to punch holes. Well, we're going to block C9 binding to our cells. So there's proteins involved in that. So we've got a lot of different ways to fend off this constant barrage of complement. C1 inhibitor promotes dissociation of C1 components. So you can kind of go through and we have expression of that. I'm going to just run through these because you can look at them later. C4, so we've got DAF. Again, these dissociation that cause these codes. So when they're separate, they don't have enzymatic activity. They're in the wrong shape, wrong conformation. So if we can break them up, they won't do damage anymore. So we've got these molecules that are involved in breaking down those C3 convertases really quickly. Factor I degrades C3b, and so it breaks them down into. So we're also going to take these guys and we're going to say, let's chew them up even more. Or we're going to have things that are called protectins, aptly named. They are going to protect our cells from C9, so basically blocking C9 from even binding to our cells. So maybe we do get this, I like to protect and inhibits the MAG attack. Okay, so you're stopping that from happening. And so we've got our cells have evolved to have these things, whereas bacterial cells do not, in general. So again, we can break things further down, and but this is where we've learned a lot. Some genetic deficiencies have been described for each of these complement components, and the outcomes may vary, but in general, patients often present with immune complex disorders due to inadequate clearance. Some may have greater frequency of infections, especially by encapsulated bacteria due to inefficient opsonization. We have animal models to study these in detail, where we can look at specific portions of the complement system. Different mechanisms exist that are highly varied. So microbes have evolved, then those that plague us have evolved ways of evading those, and some interfere with the Ig-mediated complement activation. Some may bind and inactivate portions of the complement. Some of those complement proteins, just like our cells do, they've evolved to do. Bacteria and different microbes have evolved proteases to destroy the complement proteins, or they mimic or bind complement regulatory. So they'll mimic or bind complement regulatory proteins that will help to regulate that. So some examples are For example, we've got staphylococcal protein A, removal of IgG by, so interferes with antibody complement interactions. You have S. aureus that has binding and inactivation of complement proteins. Pseudomonas has protease that mediate destruction of complement components. Streptococcus pyogenes, and variola, vaccinia viruses will mimic complement regulatory components. Again, don't need to memorize the evolution, but again, just showing that that alternative pathway evolved first, then the lectin pathway, and then this classical pathway. And actually, early on, complement served to assist in phagocytosis prior to its adaptive immunity. Just looking, if you're interested in further evolution, what animals have, what parts of these systems and how they evolved, and just our summary. Basically, complement, it's not that complicated. It serves many different purposes. Again, it's good to know those three purposes are inflammation, opsonization, and direct killing or punching holes in the cells, tightly controlled. Obviously, we need it to be tightly controlled. Provides a window into evolution of immunity. So the system was already there and worked well. Our adaptive system evolved to use those to its advantage and to be alerted by those and work in conjunction with the complement system. Understanding the system and its methods for activation and deregulation helps us better understand innate immunity evolution. Also, being able to treat, especially a lot of these disorders. I will put this up. I think I have the video. It's just a little video, I think, that shows kind of a walkthrough of the complement system. So I'll try and get that up today. So, any questions on complement? Yeah.
I found the answer for the thing that we were talking before. Is complement 11 and H. It has the ability to dissociate the C1 complement complex in order to, and it's one of those strong athletic
So they only associate it when it binds.
Yeah, it's similar to the fluid case, CDC case. It's like always like around short quantities and will associate with TO.
So they're kind of associating, dissociating, unless it stabilizes.
Exactly, with the membrane comprehension.
Yeah, cool. Okay, so I am ending there.